Hello, everyone. My name is Sanjukta Krishnagopal. I'm a postdoc at the Gatsby Computational Neuroscience Unit at UCL. And today I'll be talking about Dendritic Gated Network, a cerebellum-inspired alternative to backpropagation for learning. This work was done with the support of all these collaborators from Gatsby, UCL, DeepMind, and Imperial College London. All right. So, it is well accepted that learning in the brain happens as a consequence of change in synaptic weights. The exact mechanism underlying these changes is not very well known. Now, machine learning people will tell you that this happens through backpropagation, where some input goes in through these layers all the way to the output, where some error is computed at the output and fed back and has to go back all the way through the hidden layers. And each layer, each neuron then modifies itself, its synaptic weights, to minimize this error. Biological neurons simply don't do that. This isn't very biologically plausible. A workaround this is local learning, where the target gets fed back to each and every layer, and the job of each neuron in each layer is to predict the target directly. So it doesn't have to do any of this forward pass, back propagation business. It can instantly predict the target. And this doesn't quite work too well. But one sort of solution to this problem is something like input-dependent weight selection. And what this means is basically, here's a simple network. These are the set of weights, the weight vector connecting these set of neurons to these set of neurons in this layer. And you simply select your weights from a weight bank, which could be 2 weights or it could be n weights. And which of the weights is being used depends on the input. So for different inputs, different weights from the weight bank get used. And so a simple illustration of that is, consider a 2D input, so just x1, x2, simple. This is the entire input space. And just draw a random line across it, bifurcating the input. And in this region, if the input comes in this region, W1 is the weight that's going to be involved in learning. And if it's in this region, it'll be W2. And so each weight is only responsible for learning one section of the input, or just a subset of the input, just a smaller region of the input, which makes this do quite a bit better. And so this works, and this is wonderful. In this talk, I want to introduce dendritic gated networks, which have some of the properties of the previous slides, and also have slight architectural differences to what I described before. So this is just a regular feed-forward network. In a dendritic gated network, or DGN, each neuron, zoomed in here, has these several dendritic branches, these ones in purple. And so these branches then receive input from the previous layer. So all the neurons of the previous layer here just sort of synapse onto these different branches. And during learning, entire branches get gated on or off. So this is similar to the previous case where you had a weight bank and certain weights were chosen in one region, certain weights were chosen in another, except here you just have weights on in one region and weights off in the other region. So different branches get gated on or off. For example, this one in green here is on, these ones in red here are off. And this is actually inspired by biology, because it's known that there are biological interneurons in the brain that gate the activity of single dendrites, or these dendritic branches are inspired by these single dendrites, which are gated in the brain. All right. So how exactly does this gating happen? So each branch is turned on or off depending on the input. So let's consider this branch here in purple. Here's the gating function, this knob here that's turning the gate on or off. This is turned on or off depending on the input. So this receives the input. And the way that happens is, let's move to this square over here. This is the input space, x1, x2, just a simple input for illustrative purposes. And we draw random hyperplanes across this input space. So a random line in the 2D case. And one region, this branch is open. And if the input comes in this region, gray region here, the branch is closed. And what does a branch being open mean? It just means that weights are going through this. Weights are, this branch is allowing weights to go through it, and these weights are learning when the branch is open. Whereas when a branch is closed, those weights, nothing goes through, nothing goes in, nothing comes out. There's no learning on those branches at all. So let's consider an example here. 3 branches, B1, B2, B3. And let's consider an input that comes in this region here. So X1 and X2 have these values here. And so branch B1 is gated off. No information is going through it. It's shown in red here. Branch B2 and B3 are both gated on. They're both in the colored region. And so information is going through, and these weights are actually learning for this specific input. And now if you switch inputs, you go to a different input. B1 is gated on, B2 is off, and B3 is on. So a different subset, a different set of branches are active. And so a different subset of weights are involved in learning for different inputs. And this is key for something like this to work. So kind of putting this all together, dendritic gated networks present this novel and radically different learning rule, which is very biologically plausible, sort of derived directly from biological evidence. And it's also kind of very new in the field, both of machine learning and neuroscience. So this type of learning rule is not explored, has not been explored very much before. And the key features are input-dependent gating. So this input goes into each layer and determines whether the gates are on or off. And this leads it to be resistant to forgetting old learning. So as it's learning new tasks, it still remembers previous learning. The other feature is local learning, which makes it extremely data efficient, specifically because the loss is convex with respect to each weight. All right, so let's talk a little bit more about this resistance to forgetting. We start with a simple task here, sort of for toy task for illustrative purposes, where the input is basically this 2D input with these blue and red regions that are linearly inseparable. And the task is to separate them. And so we test this on a DGN, a Dendritic Gated Network, and an MLP of equivalent computational power, a multi-layer perceptron. And so when we do this on an MLP, first we train on task A. MLP does great. We test on task A, it does great. And these are the decision boundaries. So the MLP learns to make this entire region blue, this region red, perfectly matches the data. But then we train it on an orthogonal task, or almost orthogonal task. So we start with training on task A, then we train on task B. And then we test on task B, which is the most recent task, it does great. But then we test on task A, which is the task it has seen before. It's not the most recent one, and it doesn't do very well. So it's kind of forgotten task A already, even though it's only seen one other task. In contrast, the DGN here also train on task A, that's great. But when you train on task A, then train on task B, and then test on the previous task, task A, it remembers quite a bit. So the test A accuracy is quite high. So the DGN is much more resilient to forgetting old tasks. And so we tested this on the MNIST pixel per muted continual learning benchmark, which is more commonly used in machine learning, which is basically we had 10 tasks, each of them has different pixel permutations of MNIST digits. And without getting into too much details, you train on 10 tasks, and then you test all the way back on task number 1. And the DGN does pretty well, 70% accuracy, whereas in contrast, the MLP only does it about 50%. So I'm not getting into too many details here, but this is just to show you that this works actually quite well on also real tasks that people use to benchmark this type of stuff in machine learning. All right, so this DGN architecture and functionality is actually very much inspired by the cerebellum. And here are some reasons why. So conventionally, the cerebellum was thought to be a one-layer network, but there's ample evidence now there's actually a 2-layer network, which is these parallel fibers or PFs. That's the input. Parallel fibers go to Purkinje cells. That's layer number. These are the hidden neurons. That's the hidden layer, and then the Purkinje cells then go on to the cerebellar nuclei, and so this is the output. So this becomes a 2-layer network. These molecular layer interneurons in pink here receive parallel fiber input. So the input goes into these molecular interneurons. And these interneurons are inhibitory. So they're known to have an inhibitory effect on the dendrites of the Purkinje cells. And so in our model too, we have gating through MLIs, which is analogous to having this type of effect on the dendrites of the Purkinje cells. And lastly, the air signal is fed back in the cerebellum, is known to be fed back to not just the CBN, but also to the Purkinje cells, also to the hidden layer neurons. And so we do this as well in our model through local learning. We feed it back to the hidden layer. And so these are some of the many similarities that we have with known architectures and functionalities of the cerebellum. So we test the DGN on a cerebellar benchmark, vestibulo-ocular reflex, and is commonly used in the cerebellum community as a benchmark. It's a simple task. It's basically when an animal moves its head while focusing on an object. You move your head, your eyes move in the opposite direction. And so, you could be moving your head and the object could also be moving, in the same direction or in opposite directions. And so if it's moving the same direction, then you have what's called gain down stimulus. So your eyes have to move, your head moves X amount, your eyes have to move back less than X amount because the object is moving as well. And then if they're moving in opposite directions, your eyes have to move more. So it's a gain up stimulus, where the gain is just the ratio of head to eye velocity. And so the cerebellum is thought to be critical for VOR gain adaptation. So in this work, we map the cerebellum to the DGN. And so we use a signal, an aperiodic signal, to mimic natural head movement. This is the input. And now we directly map the cerebellum to the DGN. So we saw the cerebellum was a two-layer network. Parallel fiber input goes to Purkinje cells goes to CBN, cerebellar nuclei. And so these inputs to the parallel fibers are time-delayed versions of the signal here. And that's because different parallel fibers receive time delay, takes time for the signal to go from the retina to the parallel fiber input. And so these are time-delayed versions. They synapse onto the dendritic branches of the Purkinje cells, which then project them out to the target, to the CBN. And the target is just gain modulated eye velocity. So you wanted to tell you what the eye should do. And so the baseline we use is a multi-layer perceptron. And this is because this is the sort of only baseline in a two-layer cerebellum model. This is sort of the standard baseline, actually the only baseline. Other cerebellar baselines are restricted to one layer only. So this is a pretty obvious choice as a baseline. So this is a pretty simple task, right? You just need to learn sort of a gain modulated signal, and both the MLP and the DGN do this very well. But if you look in to see what's happening under the hood, here I plot, 5 Purkinje cells, where each Purkinje cells are the hidden layers, and each Purkinje cell has several branches coming out of it, and so each of these curves is corresponds to one branch, and each branch has several parallel fibers synapsing onto it. So along each curve, I'm plotting the weights, incoming weights from all these parallel fibers, and it's sorted on the x-axis according to the delay of the parallel fiber. So each parallel fiber has a delay. We sort according to delay, and you see these very smooth sort of patterns, smooth curves in each and every Purkinje cell. Whereas in contrast, you look at the MLP, and it doesn't quite have the smoothness. So the DGNs are learning these smooth weight distributions as a function of time delay of the Purkinje, of the parallel fiber, which is quite interesting and seems like quite natural for a biological system to do. All right. We also look into providing some experimental support for DGN. So one of the key predictions of the DGN, of the Dendritic Gated Network, is that the gating functions, which are the molecular layer interneurons, they are fixed over learning. And what I mean by gating functions is those hyperplanes that we saw. that determine whether the gate is on or off. These are fixed before learning. They don't change with learning. They're just picked right in the beginning, kind of randomly, and just fixed and kept fixed throughout learning. And so this is a prediction of the DGN model. And we wanted to test to see if the analogous system in the brain, which is the interneurons, which act as gating, they are also fixed. Their plasticity is also fixed. And so we ran an experiment of simultaneous 2-photon calcium imaging in a mouse that's learning the association between a tone cue and a reward. Credit for this experiment goes to Michael Hausser's lab at UCL and his students, Dimitar and Maxime. And the data looks beautiful, looks something like this. And we find here that, so here I'm plotting the correlation coefficient between the activity of molecular layer interneurons, or gates, and climbing fibers, or ERA. So correlation between their activity across trials. And you notice here that the gates have significantly higher correlation compared to the climbing fibers. So, as ERA is changing, so as learning is progressing, you would expect the ERA to change. And as learning is progressing, the ERA seems to be changing quite a bit. So it has low correlation across trials, but these gates seem to be relatively constant. So this is kind of consistent with our model, where we predict that the gating functions are constant, and here also the MLIs have greater consistency or more constant across trials. Of course, in experiments there's a lot of noise, so this is kind of just providing some intuition as to why our model is biologically supported as well. And that brings me to the conclusion. So we introduced these dendritic gating networks, which is a radically different and biological plausible learning rule. We propose a novel role of gating for interneurons. We show that DGNs show some resistance to forgetting, and we provide some preliminary experimental support for some of the predictions of the DGN. All right, thank you, and I'm happy to take questions.